Hello guys, good day, and here I am, Miss Merle, to talk about instrumentation in CC1. So let's start with chromatography. So chromatography is a technique for separating compounds in a mixture based on their differential affinities between a stationary phase and a mobile phase. So we've got two phases here, the stationary and the mobile phase. It allows for the rapid analysis and purification of compounds by exploiting differences in retention time and partition behavior, while ion exchange chromatography is more on it separates molecules based on their charge by using a charged stationary phase that binds oppositely charged ions. Particularly, this technique is effective in purifying proteins and other charged biomolecules by controlling pH and ionic strength. And how about mass spectrometry? So it identifies and analyzes compounds by measuring the mass to charge ratio by ionized molecules. So that's for the introduction. So let's move to the next slide. So here, what's the definition for chromatography? So it encompasses techniques used to separate complex mixtures based on different physical interactions between individual compounds and the stationary phase. So we have a mobile phase and a stationary phase. So for the key concepts in chromatography, it is a separation technique based on the differential distribution of compounds between a stationary phase and a mobile phase. So always remember we've got two phases, the mobile phase and the stationary phase. And we need to talk about also on the retention time. So this is the time a compound spends in the stationary phase before eluting or before that compound is being set free. And then we also have this thing known as partition coefficient. So this is the ratio of concentrations of a compound between mobile and stationary phases, which determines how long a compound is retained. So for the components of chromatography, we've got four. We have the mobile phase, the stationary phase, the column, and the eluate or eluate. So the mobile phase, guys, is this is the fluid or liquid part, and it moves. This is the part that moves through the stationary phase, and it carries the sample components with it. So the movement of the mobile phase allow for the separation of compounds based on their interactions with the stationary phase. So how about the stationary phase? So guys, for the stationary phase, it is the material that remains fixed, hence stationary, right? Inside the chromatography system, and then it interacts with the sample components, causing them to separate as they move through the stationary phase at different speeds. And then the column is the container, it holds the stationary phase. Okay, it's the container, or we can say also that it serves as the tube that holds the stationary phase. So in most chromatographic techniques, the sample and the mobile phase are passed through the column, and it is in the column where separation occurs based on the interaction between sorry guys, I'm really having a cough, between the sample components and the stationary phase. And lastly, we have the eluate or eluate. So it refers to the separated components. So the eluate is the fluid that exits the chromatography column, and it contains the separated components of the sample. And as the compounds are eluted at different times, they can be collected and analyzed separately. So again, for the components, we've got the mobile phase, the stationary phase, the column, and the elute. So next, we've got the modes of separation, and we've got four, starting with liquid to solid chromatography. We had It's also known as adsorption chromatography. We have liquid to liquid chromatography, partition chromatography, steric exclusion or size exclusion chromatography, and lastly, ion exchange chromatography. So for our adsorption chromatography, liquid to solid chromatography or LSC, this is a type of chromatography where the separation of compounds is based on their absorption onto a solid stationary phase. So in this technique, we have the stationary phases, or the stationary phase is it is a solid material, guys. Sorry. So silica or alumina and these solid materials absorbs responsible for the absorption of the components of the sample. And then the mobile phase is a liquid, of course, liquid, so mobile. And it flows over the stationary phases or phase, carrying the sample. And as the mobile phase moves through the stationary phase, different components, guys, are being, how to say, they are adsorbed onto the solid surface to varying degrees. So components with stronger interactions with the stationary phase, they move more slowly, while those with weaker interactions with our stationary phases, they move faster. So it allows for the separation of sorry guys, allows for the separation of compounds based on their absorption affinities. And for the usage, well, liquid LSC is not widely used in clinical labs due to challenges with achieving homogeneous distribution of adsorption sites. So that's with LSC. Now let's move to LLC, or liquid to liquid chromatography. So this is a separation based on the partitioning of compounds between two immiscible, so we have polar and nonpolar, two immiscible liquid phases. So in this method, guys, what happens, the stationary phase is a liquid that is immobilized on a solid support within the column. So the mobile phase is another liquid that how can I describe it, it's another liquid that flows over the stationary phase. So separation occurs as sample components distribute between the mobile and stationary phases based on their relative solubility. So, guys, compounds with a higher affinity for the stationary phase, they move more slowly, and the opposite happens for those lesser affinity to that of the stationary phase. So here, for the normal phase, mobile solvent is less polar than the stationary solvent, and the reverse phase, the mobile solvent is more polar. So we are talking about two immiscible samples or solvents here. We have the polar and the non-polar, polar solvent. And then these LLC is best for non-ionic compounds. So that's for LLC. And then now we have steric exclusion or size exclusion. That's the other name for steric exclusion chromatography. So it is a separation technique that is based on, yeah, it separates molecules based on their sizes and their shape. So here, the stationary phase is made up of porous beads or gel, and the mobile phase is a liquid. Always remember, guys, the mobile phase is it is liquid in state, and then it carries the sample through the stationary phase. And here, the packing materials, usually hydrophilic beads, while modern methods use inorganic materials like silicates or silica or glass. So largely, applications for steric exclusion or size exclusion chromatography is for separating molecules based on size, and with variations such as gel filtration and gel permeation chromatography. So next, guys, sorry. We also have ion exchange chromatography, or IEC. So this is a subtype of chromatography that involves the separation of molecules based on their charge, based on their charge. So in an IEC, so charged stationary phase interacts with oppositely charged analytes in the sample. So here we have a cation exchange or cation exchanger. So what happens there? There's binding of positively charged ions, and the anion exchanger can bind to negatively charged ions. So later, there's a diagram for that. And then commonly, IEC is utilized for protein separation, nucleotide separation, and other biomolecules. And it is really effective in purifying biological samples. So that's IEC. So separation of molecules based on their charge. So here, as what we can see for IEC, so positively charged protein binds to negatively charged beads. So these are the beads, negatively charged beads, the gray ones. And then the proteins bearing much positive charges are bound to the negatively charged beads. While in contrast, the negatively charged protein, see, less of the positive but more of the negative charges, they just easily flow through the column. And then here, so in this process, guys, for in our IEC, so we have two types of exchangers earlier. I have mentioned this one. So we have the cationic and our anionic exchangers. So we can use both. In cationic exchangers, they possess negatively charged group, and these will attract positively charged cations. So these exchangers are also called acidic ion exchange materials because of their negative charges result from the ionization of acidic groups. And then the opposite is our anion exchange or stationary phase particle or anion exchanger. Sorry. So our anionic exchangers are they have positively charged groups, as you can see here. And these positively charged groups will attract negatively charged anions. So these are also called basic ion exchangers, basic ion exchange materials. So ion exchange chromatography is most often performed in the form of column chromatography. However, guys, there is also TLC methods that work basically based on the principle of ion exchange. So that's it. And then, I'm sorry, guys. So here, ion exchange chromatography. So mechanism is more on, it's based on the magnitude and charge of ionic species. So this is just trying to be in detail with the previous slide, no? So hot ion exchange resins. So resins with sulfonate groups exchange hydrogen ions for cations like sodium. And then anion exchange resins with functional groups that like diethyl amine exchange with OH ions for anions like chloride. And then applications for if we wanted to remove substances that interfere in our samples, we can do IEC. And we can also do dilution of ion solutions by concentrating them and mixture separation of charged molecules like amino acids. So all those things will call for IEC as the test or the methodology. And then adjustments. So to facilitate separation, the pH and ionic concentration of the mobile phase can be altered in our IEC, guys. Next, so how important chromatography is in the clinical laboratory. So basically, these tests are utilized for, especially when they are coupled with mass spectrometry. So chromatography is LCMS or GCMS is considered the gold standard of many tests because of it offers high sensitivity and specificity. And the increasing variety of analytes in clinical setting has really driven the demand for advanced chromatographic technologies. But it's not much. In the Philippines, we can see these machines are more in the research setting and not much in the clinical laboratory, actually. And then we have three chromatographic procedures. We have TLC for thin layer chromatography. We have HPLC. So sorry, guys. We have high performance liquid chromatography, and we also have gas chromatography or GC. So for our high precision or high performance liquid chromatography, this one is really an advanced form of liquid chromatography, and it's used for the separation and identification and quantification of components in a mixture. And of course, there is also this how do we call this thing, guys? The mobile and the stationary phases. So the mobile phase is a liquid solvent here or a mixture of solvents, and it's pumped through the system under high pressure. So again, the mobile phase is a liquid solvent. So the mobile phase, the blue one. And then the stationary phase is typically a packed column. So we have a column here. This is the stationary phase. This is the mobile phase. The mobile phase is a liquid solvent, while the stationary phase is typically a packed column with small particle sizes that provide a large surface area for interactions. And then, guys, as the sample passes through the column, so different components separate based on their interactions with the stationary phase and their solubility in the mobile phase, of course. And sorry. HPLC is widely used in pharmaceuticals and environmental analysis and food safety because HPLC offers precision, speed, and the ability to handle complex mixtures. So common in actually in research labs again. And then for our high precision liquid chromatography components, we have the pumps, the columns, sample injectors, detectors, and recorders. So the pump, so it's a mechanical way of reciprocating pump. No, no, they are most commonly mechanical reciproc. Sorry, guys. Mechanical reciprocating pumps are most commonly used or utilized because they offer constant flow. We also have stainless steel columns packed with silicates or silica, and in it's also popular the reversed phase HPLC with nonpolar stationary phases. For the sample injectors, or specifically the loop injectors, they are used for high reproducibility to introduce sample into the mobile phase. And detectors, we've got a lot of detectors that can be utilized. We have the UV visible spectrophotometers, the photodiode arrays or PDAs, fluorescence, electrochemical detectors. They are commonly used for detection. And then recorders to record detector signals versus time, so that we can have the chromatogram. So this is the endpoint of our technique. The form once we will print, there's a printout that's called as the chromatogram. And then retention time and peak area are also used for compound identification and quantification. So we will know how long a particular sample remains in our HPLC machine and the peak area that it can produce, because different compounds and different yeah, they have different retention times and peaks. They develop they manifest different peaks. So here, guys, so the components of our HPLC in detail, we have the, where is our pump, solvent delivery pump. So the pumps in HPLC are responsible for delivering the mobile phase at high pressure. Take note, at high pressure through the system and the column to reach, so that our sample can reach our column. So we needed this pump. And then the pumps ensure a constant flow rate and can be adjusted for different types of analysis. So this high pressure delivery is the key to efficient separation. So the pump has this utilizes high pressure to deliver the sample. And then for our columns, where's the column here? So the column is packed with a stationary phase, usually silica based, guys. Silica based, and it is where the separation of compounds occurs or happens. So, depending on the stationary phase material, the column can be used for different types of separation, such as reverse phase or ion exchange. And then our next, followed by our sample injector. So the sample injector introduces the sample into the mobile phase stream with high precision, and this can be done manually. So you can inject manually the sample or through an automated system. But for me in Bangkok, I don't do the manual thing. I rely on the automated system. So this is to ensure the sample is delivered in a small, accurate volume. And then we also have the detector. So the detector is responsible for monitoring the separated compounds as they elute from the column. So common detectors include again the UV fluorescence or mass spectrometers. And then because those things measure different properties like absorbance or mass, so they tend to help identify and quantify the analytes. And we also have the last part is the recorder, so that we can have our chromatogram, because the recorder produces after from the detector to the recorder, then the end point is the production, the printout, that's the chromatogram. So the recorder or data system, it captures and processes the detector signal, producing a chromatogram. So the chromatogram is a graphical output, guys, that shows the retention times and the intensities of the separated compounds. So, what do you mean by retention time? What is that? The peak miss we wanted to see. So, in the next parts, we can see those things I'm talking about. So we also have illusion techniques in HPLC. So these illusion techniques refer to the methods used to move and separate compounds through the column using the mobile phase. And the main illusion techniques, we have the isocratic illusion and the gradient illusion. So for the isocratic illusion, the mobile phase composition is constant, while um, opposite to this is that the composition of the mobile phase in our gradient illusion, it changes over time. So this one is constant, this one keeps on changing, the gradient illusion. So here, guys, so what is gas chromatography? So it is an analytical technique used to separate and analyze compounds that can be vaporized without decomposition, or it separates volatile compounds or compounds that can be made volatile. Okay, so we also have the mobile phase. The mobile phase, guys, is the carrier gas. Carrier gas, an example would be helium or nitrogen, and then that carrier gas serving as the mobile phase will carry the sample, guys, will carry the sample through the column. We also have a stationary phase. So what is the stationary phase? Usually, it's coated or packed. It's a coated or a packed. This one, the stationary phase is sometimes it's coated or it contains a liquid or solid solid stationary phase that interacts with the sample components. So it could be liquid or solid. Here we can say that it's solid. And as the sample is injected, guys, as the sample is injected into the heated column, this column is heated. Here, as we can observe, there's an oven. Its components are separated based on their volatility and interactions with the stationary phase. See? And then what happens next, miss? So compounds with lower boiling points elute first. Again, compounds with lower boiling points elute first for a while. While those with higher boiling points, guys, they elute later. So GC is commonly used for analyzing gases, volatile compounds, and liquids that can be easily vaporized. So that's the main significance for using gas chromatography. So compounds which are volatile or compounds that can be made volatile. So here, going back to the components, we have the carrier gas or the mobile phase. This is what I have said earlier. So nitrogen, helium, in addition is argon. And then next, sample injection. So samples are injected as gases or vaporized on injection. And the sample vapor is swept through the column with components separating based on volatility. And columns, GC columns, guys, gas chromatography columns are often made of glass or stainless steel, or either packed or capillary. And then the stationary phase is a non-volatile liquid, and separation is based on volatility or polarity. So we also have the detectors, two types of detectors for GC. So thermal conductivity detectors, they measure changes in electrical resistance. So due to changes in temperature changes as the sample passes through. And we also have FID or flame ionization detectors. These detectors are they offer high sensitivity. They can measure ions produced when the sample burns in a hydrogen flame. And then gas chromatography chromatograms is this is the readout or the printout. So we can see really it's used to identify and quantify compounds by retention time and peak area. So the retention time in the x-axis and in the y-axis we have the response. And we can observe the peak width and the peak height. So peak width is measured at half the height of the uh, compound. And then guys, the peak area is this one, this is the peak height, and this is the peak width. So different compounds should manifest or show different peak heights. See, for hydrogen, almost insignificant, but for carbon dioxide, is very the peak height is very high, and we can also the retention time or the yeah, it varies from one compound to another because, for example, your with oxygen here like 6, but it's 14 for CO2 or carbon dioxide. So that's a way to interpret or read the chromatograms. So here. Again, y-axis showing peak width, peak height, but for our retention time, we can really see the differences from one compound to that of another. So the summary of key points for TLC for good for quick screening and qualitative. While if you wanted something with higher precision and quantitative, HPLC and GC are meant for quantitative analysis. GC is ideal for volatile compounds and it is highly sensitive with advanced detectors like TC and FID. While for HPLC, guys, if we are dealing with liquid samples, so HPLC permits ionic, non-ionic, and ionizable compound separation. So for mass spectrometry, this one is an analytical technique used to identify and quantify compounds by measuring the mass to charge ratio of ions. So it has four key steps: ionization, wherein the compounds are ionized to form charged particles or ions in an ionization source. So, in what way, miss? There is this electrospray ionization and Maldi-Toff ionization. So, ionization, that's the first mass analysis, second step, and there is detection for our sorry, mass spectrometry. So, key components, we have our ionization source. So this one is responsible for the conversion of molecules into ions. So we will be using ESI here. Our pre ionization. And then mass analyzers separates ions by their mass to charge ratio. So here comes our time of flight or TOF and the quadruple. And for the detectors or detector, they are responsible for recording ion signals to generate a mass spectrum. So always remember the key components and how what is the main principle of a particular technique or method. So for this one, it involves the measurement of mass to charge ratio of the ions. So what do you mean by mass to charge ratio, guys? In mass spectrometry, so it is a representation, or it represents the ratio of the mass of an ion to its charge. Again, the ratio of the mass of an ion to each sorry charge. So it is a critical parameter used to identify and analyze ions. So here, guys, mass or M stands for mass, and it refers to the mass of the ion. So typically measured in AMU or Dalton. So we have two ways to measure the mass of the ion: atomic mass units or by using Daltons. And we also have Z for charge. So this represents the number of elementary charges on the ion, which can either be negative or positive. So, for example, guys, it's very easy to know the MZ ratio of a particular ion. So, if an ion has a mass of 100 AMU or atomic mass units and a single positive charge, it carries a single positive charge. So, you will just divide 100, its AMU by 1 with its single positive charge, only one positive charge. So, the answer will be 100. So what if that same ion having the same mass of 100 atomic mass units, and that ion is doubly charged, so plus 2. So 100 divided by 2, that will become 50. And then here, so mass spectrometry, so we're going back again to remember. So it's used to identify compounds. Yeah, with all these things in instrumentation for chemical chemistry, it's more on identifying compounds, and the way of identifying them truly vary. So varies. And then here for MS, it's based on their again MZ or the mass to charge ratio. So whenever our MS is coupled with, like for example, methods like GC. So it becomes GCMS or HPLCMS. So this really allows definitive identification of the components in complex mixtures. That's why the instrumentation in CC is largely used in forensics or forensic analysis. So this is a block diagram that shows the basic parts of a mass spectrometer. So the inlet transfers the sample into the vacuum system, no? Into the vacuum of the mass spectrometer. So in the source region, neutral sample molecules are ionized, and then they are, how to say this one, they are accelerated into the mass analyzer. And the mass analyzer, this one, guys, is the heart of the mass spectrometer. This section is really capable of separating ions either in space or in time according to their mass to charge ratio. And the ions are separated, they are detected, and the signal is transferred to the data system for analysis. And all mass spectrometers also have a vacuum system to maintain the low pressure, which is called high vacuum, required for the operation. And why a high vacuum is required? So high vacuum minimizes ion molecule reactions, scattering and neutralization of the ions. So in some experiments, the pressure in the source region or a part of the mass spectrum photometer is intentionally increased to study these ion molecule interactions or reactions. So under normal operations, guys, however, any collisions will interfere with the analysis. So again, don't forget the mass analyzer is the heart of our mass spectrometer. So this one, see in this technique, guys, the molecules are bombarded with a beam of energetic electrons, so because through the electron beam, and then the molecules are ionized, sorry, and broken up into many fragments, some of which are positive ions, and then each kind of ion has a particular ratio of mass to charge, so Me ratio. And then for most ions, the charge is 1, and thus the Me ratio is simply the molecular mass of the ion. And then, guys, the ions pass through the magnetic and electric fields. So there is this magnetic field or electric fields to reach the detector. So before the ions can reach the detector, there's a need for them to pass through a magnetic field or an electric field. So in the detector, this is where the ions are detected and signals are recorded to give mass spectra, hence mass spectrometry. So for mass spectrometry, we have the basic components: sample inlet, ionization source, mass analyzer, and the ion detector. So responsible for introducing the sample into the MS, that's our sample inlet, and the conversion of our molecules into ions, so ionization source, and then separation of the ions this time based on the relative mass to charge ratio. So that's the function of the mass analyzer. And for the measurement of the intensity of the ion current for each mass to charge ratio, that's the role of our ion detectors. Always remember, our ions before reaching the detector, they need to pass through that magnetic or electric field. And then here, guys, we have two types of ionization techniques for mass spectrometry. We have electron ionization or EI and our atmospheric pressure ionization API, under which are two examples. We have electrospray ionization SC and atmospheric pressure chemical ionization APC. Sorry. So here for our EI, or electric ionization. This is a common ionization technique, guys, actually, and used in mass spec, and especially for analyzing for small to medium-sized molecules. So the process involves there is this sample introduction. So the sample is introduced into the ionization chamber, usually in a gaseous form, and then there is it is followed by what, guys? By an electron bombardment. So the sample is bombarded with high energy electrons, typically 70 eV, and generated by, and this high energy electron is generated by an electron source. And then ion formation. So again, sample introduction, electron bombardment, and ion formation. So ion formation, guys, the high energy electrons collide with the sample molecules, causing them to lose an electron and form positively charged cations. So in this process, uh, no, this process also results in the fragmentation of the molecule into smaller ions. And see, so fragmentation patterns are reproducible and they allow or permit comparison with the reference spectra. So if we wanted to identify volatile molecules but very small, then we need to use electron ionization. Next, we also have the still under ionization techniques, our atmospheric pressure ionization, and specifically atmospheric pressure chemical ionization. So is an ionization technique that is used in mass spectrometry, particularly for if we are talking about liquid samples, guys. So we have, unlike SC, APC involves vaporizing the liquid sample in a heated vaporizer before ionization. And then by using a corona discharge needle, because this one is capable of emitting electrons, and this electron emission is able to ionize the compounds in the gas phase. So the advantages is particularly useful for less polar compounds and complements easy ESI in LCMS. So this one again, if we are trying to analyze liquid samples, so atmospheric pressure ionization, specifically atmospheric pressure chemical ionization or APSI. And then for the mass analyzers, for mass spectrometry, we have two types of analyzers, quadrupole and ion trap. So quadrupole mass analyzer. So we can describe this one as a quadrupole mass analyzer consisting of four parallel rods arranged in a square configuration with alternating voltages applied to the rods. So four parallel rods with alternating voltages applied to the rods. And then for the ion trap mass analyzer, we can describe this one as this is a device that uses electric or magnetic fields. So ions, see? Electric or magnetic fields to trap ions in a confined space. So trapping ions in a confined space, that's for our ion trap mass analyzer. And if we're talking about parallel rods arranged in square configuration with alternating voltages applied to the rods, that's our quadruple. Quad, see? Four parallel rods, quadruple. Okay? It's easy to remember that. And then here, in detail, quadruple, so consists of four parallel rods that create an oscillating electric field. So here, only ions with specific mass to charge ratio can pass through the detector. And we have two modes. We have the full scan mode and the selected ion monitoring or SIM or SIM. So if the scanning is within a range of the mass to charge values, providing a complete mass spectrum, that's a full scan. But if there is the specific focusing on the specific mass to charge values with increasing sensitivity for the compounds of interest, then we're talking about selected ion monitoring or SIM. So again, this one, complete mass spectrum, complete, that's your full scan. But we are trying to specify the mass to charge values, then that's SIM or selected ion monitoring. And then the ion trap in detail. So this one is a modified quadruple, still a quadruple, guys, but it can trap and store ions. See, ion trap, trapping, storing ions, ion trap. And ions are selectively ejected based on the differences on their mass to charge ratios. So for the types, we have the linear ion trap and the 3D ion trap, two types under ion trap. So if the ions are confined along the axis of the quadruples, that's linear. But however, if spherical trap for ions are being formed, that's your 3D ion trap. And then the advantages would be ion traps can accumulate ions over time. So there is this resulting to an enhancement when it comes to sensitivity for low abundance compounds. Next, we have the mass spectral analysis. So, if you can read the phrase mass spectrometry or mass spectral analysis, always think of the phrase of fragmentation patterns because fragmentation patterns in mass spec are crucial for several reasons like structural elucidation. So fragmentation patterns provide detailed information about the structure of the original molecule, guys. So by analyzing how the molecules break apart into smaller ions, of course, scientists can deduce the arrangement of atoms and functional groups within the molecule. Second, why is it fragmentation patterns are important? Because we can do compound identification, and this is really relevant in forensic analysis because unique fragmentation patterns serve as fingerprints for specific compounds. So if we will do compare we will comparing these patterns to non-reference spectra helps in the identification of unknown substances. So yeah, guys, especially for forensic analysis. And third, we have quantitative. So we can do quantitative analysis on the fragmentation patterns. So we can use fragmentation patterns to monitor specific ions related to the target analyte. Or if we wanted to improve the accuracy and specificity of quantitative measurements, so it's really important to know fragmentation patterns. And lastly, we can also confirm molecular identities, guys. So the fragmentation pattern helps really confirm the identity of a compound by providing complementary information to the molecular ion peak. So this is particularly useful for verifying the presence of a compound in a complex mixture. So that's it, guys, for our mass spectral analysis and focusing on the fragmentation patterns. So summary of key concepts. So MS coupled with GC or LC, or gas chromatography, and liquid chromatography. They are really useful for the ID of compounds, definitive, take note, definitive identification of compounds. And we also have ionization methods like ESC and APC. So they are chosen based on the sample type and desired sensitivity of the test. You also have ionizers in the form of ion traps and quadruples. So they both provide different advantages depending on the analysis and whether broad screening or targeted analysis is we are trying to achieve. So the reproducibility of EI in GCMS allows for reliable ID using reference spectra, while the flexibility of ESC and APC allows the analysis of a wide range of compounds, including large biomolecules. So, what is tandem mass spectrometry? So, MS. So, here, this is a technique where two or more mass analyzers are utilized in sequence to give enhanced analytical capabilities. Typically for identifying and characterizing molecules like peptides, proteins, or small organic compounds. And this one, it enhances the capabilities of our conventional mass spec by allowing greater selectivity and lower detection limits. So this technique is commonly implemented in using instruments like our triple quadruple MS, and often utilized in both GC-MSMS, and we have LC-MSMS. So MSMS, tandem mass spectrometry. So again, using two or more mass analyzers in sequence to provide a detailed analysis of ions. And initially, so we have a precursor ion, and then it is selected and fragmented in a collision cell, and the resulting fragment ions are analyzed by a second mass analyzer. So we have two analyzers here, hence tandem mass spectrometry, MSMS. So again, we have the first mass analyzer, and then the second mass analyzer is responsible for analyzing the fragmented ions. Okay, and then next, so we have tandem mass spectrometry. We have triple quadruple mass spectrometer, the triple Q, C, Q, Q, Q. We also have scanning modes in triple quadruple MS and a high resolution mass spectrometry, which is widely used in research nowadays, specifically MALDI-TOF. So here, quite lengthy, sorry. So triple quadruple mass spectrometer. So it has three, triple of course, three quadruples arranged in series, and each quadruple has a distinct or different function. So for Q1 or quadruple one, so it's responsible for scanning the presets of the mass to charge range so that the specific ion of interest or the precursor ion or parent ion can be selected. So if we are thinking about what will scan the mass to charge range, that's the role of our Q1. And then if we are talking about something that functions as a collision cell, where the selected parent ion or precursor ions collide with the neutral gas molecules in the form of argon, helium, or nitrogen so that we can achieve fragmentation. So we need that Q2 or quadruple 2. So actually, this process is known as your collision induced dissociation. So seed will produce the daughter ions. So at first, we're looking for the parent. The parent ions are achieved in Q1 and the daughter ions are in Q2. So, how about Q3? What's the function of Q3? Q3. So it's on the analysis of the product ions generated in our Q2. The product or the daughter ions are analyzed in our Q3. Okay? And Q3 can either scan all the product ions to produce a full product or selective. So it can scan everything or it can do scanning on a particular ion. So that's the thing with our triple quadruple mass spectrometer. Scanning modes in triple quadruple MS or mass spec. So we have the full scan MS. So all ions across the master range are detected. We also have SIM. So it focuses only on particular sets of ions. And then product ion scan. So selects ions of one master charge ratio in Q1, fragments them in Q2, and allows all fragments to pass to the detector, providing a detailed fragmentation pattern. We also have SRM for the selected reaction monitoring. It's similar to the product ion scan, but only specific fragments of selected master charge ratio values are allowed through the detector. So high resolution MS, we have two types, time of flight and orbit trap. So high resolution mass spec. So it gives greater accuracy in determining the mass of compounds, especially for complex biological matrices and drug screening. So if we wanted to do analysis and our samples are for drug screening or really complex in nature, then we can utilize time of flight and orbit trap. So time of flight. So here, lighter ions travel faster than heavier ions. And then by measuring the time taken for an ion to traverse the flight tube, we can determine the mass to charge ratio. So the time it takes for taken for an ion to traverse the flight tube. This is the flight tube starting from this part. And then for the resolution, it can typically achieve resolutions of 10,000 to 50,000 full width at half maximum. So this is really an accurate determination of the mass, guys, with our high resolution time of flight assays under tandem mass spec. We also have orbit trap. So here, the ions are injected tangentially to an electric field between a barrel outer electrode and a spindle-like inner electrode. So, better look, the spindle-like inner electrode. So the stable orbit achieved by the higher mass to charge ratio ions allows for accurate mass measurement. And then take note, it offers a higher resolution as compared to our time of flight. So here, this time is 100,000 to 250,000 as compared to that of the only 10,000 to 50,000 full width at half maximum for our time of flight. So our orbit trap provides very high precision. And then guys, next is we have our tandem mass spec detectors. So for the detector, we have the electron multiplier. And this is the most common detector used in mass spec. So the electron multiplier is responsible for amplifying the signal generated by ions. So the green ones are the signal generated by the ions, and then it consists of a series of dinodes. These are the individual dinodes, guys, and the dinodes are responsible for amplification or amplifying the signals generated by the ions. So when the ions strike the first dynode, the electrons are emitted and are subsequently amplified through a cascade effect across the dinodes, resulting in a significant signal amplification. So in the end, once it's in the collector plate or detector plate, there is a significant signal amplification already in our electron multiplier. So it's a series of dinodes, and then there is this cascade effect across the dinodes when it comes to the signals generated by the ions. So summary of key concepts. So for tandem MS, especially using a triple quadruple setup, so it allows for highly selective and sensitive analysis by ion fragmentation. We also have high resolution MS technologies like time of flight and orbit trap, which enhances mass accuracy. And for if we are dealing with very complex samples, we can utilize high resolution MS in the form of our TOF or orbit trap. But the one which offers a very high precision is the orbit trap. But having TOF is already considered okay. And scanning modes in MSMS, so it provides flexibility for both a comprehensive screening and targeted compound analysis, while the electron multiplier is a critical component in detecting and amplifying the ion signal. So applications of mass spec MS, guys, is really versatile in nature, and hence it's widely used in clinical laboratories for the identification, quantification, structural analysis of small molecules, proteins, and pathogens. But especially for we can maybe observe this abroad and in tertiary laboratories. So its applications span toxicology, drug monitoring, proteomics, and even point of care diagnostics. So we have the small molecule analysis, GCMS and LCMS for drug testing. So GCMS is employed largely in urine toxicology if we wanted to confirm the presence of drugs of abuse. So the process involves drug extraction or extraction of the metabolites from the body fluid, in this case urine, and then we will react them with derivatizing agents to make them more volatile and analyzing them using the libraries for compound detection. Okay, so that's for our GCMS or gas chromatography mass spectrometry. We also have LCMS. So if we are talking about polar and heat labile compounds without extensive extraction or derivatization. So it's time for us to utilize LCMS. And this one of a few, no, not one, but few of the advantages it can offer is that it is time and cost efficient, and then chromatographic separation can be less robust though, so that's a disadvantage, so leading to wider peaks and variable retention times. And then applications beyond toxicology for our LCMS. So if there are some analytes, guys, which are having a very low concentration in our body fluids, like vitamin D, testosterone, and if we are looking for like drugs, specifically immunosuppressant drugs, then using LCMS, this one offers superior sensitivity and specificity over the traditional immunoassays we're doing in serology. And then, however, one disadvantage of LCMS is that ion suppression is it's affected by ion suppression, and it really needs skilled operators to how to say to use the machine. And then we have high resolution MS, so used for unknown toxicology analysis and identification of designer drugs. So with soft ionization, so high resolution MS can differentiate compounds by their exact molecular weights. We also have UPLC and multiplexing. So ultra performance liquid chromatography. So this one permits faster flow rates and reduces runtimes by handling high pressures. So if we are doing high volume testing, that's the time that your lab or the lab that you're working must use UPLC or ultra performance liquid chromatography. You also have multiplexing. So multiplexing is on if we wanted connecting multiple LC pumps to a single mass spectrometer. So it reduces analysis time by staggering the flow from each pump to align with the mass. And then this method is for high throughput targeted analysis. So high volume testing, high throughput, think of UPLC and multiplexing. And here comes proteomics. So, proteomics, what do you mean by that? So, it is the large scale study of proteins. So, particularly their functions of proteins are being studied, the structures of proteins and interactions within the biological system. So, usually proteomics, guys, involves identifying and quantifying proteins in a sample, analyzing their modifications and interactions, and understanding their roles in cellular processes and diseases. So, proteomics provides insights into the dynamic changes in protein expression and function, which are crucial for understanding biological mechanisms, and so that we can develop new diagnostic and therapeutic approaches. Okay. So we have techniques like 2D electrophoresis and Malditov, our matrix assisted laser desorption ionization, time of flight, are used to identify and study proteins, including post translational modifications of proteins. So that's with our Malditov. So here again, matrix assisted laser desorption ionization, time of flight. So this one, it is a mass spectrometry technique used for analyzing biomolecules and other large compounds. And then the application is for analyzing, see, large compounds, so peptides and proteins by desorbing and ionizing samples with a laser pulse. So the technique is very essential for identifying proteins and assessing their molecular weights. And again, protein post-translational modifications can be achieved. We can detect these modifications by using MALDI-TOF. And to connect this in with the clinical lab, so pathogen identification using MALDI-TOF, it can be used for bacterial and fungal species identification by generating a protein fingerprint from ribosomal proteins. And the good thing, actually, this method is really cheaper and faster as compared to our biochemical techniques. And it can really give rapid identification of that pathogen directly from cultured colonies. So those are the advantages of MALDI-TOF, guys. Matrix assisted laser desorption ionization, time of flight. We also have PCR with electrospray ionization, time of flight, MS. So if we wanted to identify pathogens directly from the patient samples, and then how is the identification being made or done? So it's by amplifying DNA by using PCR, so that the exact masses of the DNA fragments can be computed or detected. So this one is the presence of PCR ESI TOF, guys. It can really eliminate the need for culturing organisms. So in the future, that time might come that we will no longer there's no longer that need for us to do uh, cultures for organisms. Rather, we will just use PCR ESITOF because we can directly identify pathogens directly from patient specimens with this technique. So that's all, folks. So enjoy and have a great afternoon.